Okay, so today this one is Cellular Respiration Part 2. I'm going to show you oxidative decarboxylation, and I'm going to show you the Kreb cycle, also known as the citric acid cycle. Alright, when we ended with larger organisms having oxygen to bring the biochemical pathway of cellular respiration on down the path, we stopped with pyruvic acid. Remember that pyruvic is a 3-carbon molecule. Okay? What the deal is at this point, we cannot continue on. We're knocking at the door of the mitochondria. And so the mitochondrial membrane is basically right about here. This could happen either out here or in here. What's going to happen is that NAD goes into the equation. Should be an arrow really right there. And it's going to grab some hydrogen, which of course we know at this point is that we're putting energy into that bond right there, and that we're also making something happen to this 3-carbon molecule here. In this case, CO2 is kicked out. Now that's what you're going to breathe out. Anyway, so we have now at this point what's something called an acetyl group, and this is a 2-carbon molecule. And in your book and on the green board, this 2-carbon molecule is going to hook up with something called coenzyme A. Coenzyme A is very important. It's vitamin A. And what the deal is that coenzyme A is something that is stored in your tissues and is always there. It doesn't get flushed out with anything else. That's why vitamin A can be toxic if you take too much. But anyway, so we have 2-carbon molecule, and we're going to hook CoA on it, and this is called acetyl-CoA. It enters the Krebs cycle, and at this point we are in the matrix, which is the fluid of the mitochondria. It enters the Krebs cycle, and it hooks up to this big, huge name right here, which you don't have to know, but it's a 4-carbon molecule. Now, my comment would be 2 carbons plus 4 carbons gives us 6 carbons. So that's where we stand right now. This name, you want to remember, that's where it gets the name citric acid cycle. Alright, now you see all these other names, and they're in your book. You do not have to know these. But what you do have to know is the fact that this is occurring and this is occurring. Now, this is, at this point, a 6-carbon molecule. When you kick out that CO2, this is a 5-carbon molecule. Alright, you're going to do the same thing again. You're going to kick out a CO2 because NAD goes in and grabs this hydrogen stuff until you eventually get to what is called a 4-carbon molecule. This one is 4-carbon. This one is 4-carbon. This one is, of course, 4-carbon. We've already said that. And what the whole point of it from here to here is that you literally are changing this back into a usable product that can now enter the cycle again. But there's a little bit more. So here's what we've done. We've gotten our 6-carbon. We have now kicked out 2 carbons by using getting rid of CO2. We are back to our 4-carbon. At this point, recognize that the 3 carbons from what you started with, you had 3 carbons for this Krebs cycle, you had 3 carbons for the other Krebs cycle. That's your 6-carbon glucose that originally came into the cell. At this point, right here, it's all gone. So now we're going to try and eke out just a little bit more energy. And we do that by taking another energy carrier, and it's going to grab some hydrogen. And we're going to put an energy in that bond right there. Alright, at this point, we're also going to do the same thing again by introducing another NAD, grab some more. And by making all this hydrogen come out, we're of course changing the shape or the conformation of this molecule in order to turn it back into that molecule so it can grab the next acetyl-CoA, make citric acid, and start the whole cycle over. There's one thing missing from here, and this kind of bugs me. I'm going to put it right here. It doesn't really matter. But also at this point, ADP is going to enter. I swear it's ADP, and ATP comes out. I'm getting better. Alright, so let me remind you, I'm going to go up here to the top of the page. We had to the good 2 ATP from glycolysis. To the good. Remember, we used 2 and made 4, but that's only 2 to the good. We also made, during that pathway, 2 NADHs. You'll notice down here they have NADH plus H. Some of your books will actually have NADH plus, 2. Doesn't matter, just remember that you grabbed them. We also here have made this NADH, and we made it on the other side also, so there's plus 2 NADHs made right here in oxidative decarboxylation. That's what this is. For those of you that had me in honors biology, if you'll remember, that was a bonus point that you had to remember that you took out a carboxyl group. Alright, so we've made the NADH here, we've made all this stuff up here. What we've done in this cycle is we have made, and how do I get rid of that? Okay, no, stay right there. Got it. Alright, what we've done so far in this cycle, we made one here. Let me get a different color. We made one here. Alright, we made one here. We made one here. So right now I'm going to write down 3. Alright, we've made another color. We'll see if yellow shows up pretty decent. Nope, doesn't show up. Hang on a second. Alright, we've made this right here. So I'm going to write down 1. And we also made that right there, the ATP. So I'm writing down 1. So what we have is we've made 3 NADH. We've made 1 FADH, 2. And we've made 1 ATP. However, remember, this is all times 2. Sorry, times 2. So our total for the Krebs cycle, lost my pen, is 6. Alright, I'm going to pause this thing if I can figure out how to do it. Back in a second. So what we've made for both Krebs cycles, we had 2 sets of 3 carbon molecules come in. We have made 3 NADH. We've made times 2 FADH2s. And we've made 2 ATPs. Alright, last thing I'm going to do, we'll try and get a lighter color. Let's try a hot pink. Alright, I want you to look. I'm going to circle what you need to know. There's the amount of things we made from glycolysis. Alright, this is what we made from oxidative decarboxylation. And this is what we made from the Krebs cycle or the citric acid cycle. Those are the things that you're going to need to know. Very quickly, watch the pointer. We took a 3 carbon molecule up here, got rid of a CO2. That's a 2 carbon molecule called acetyl-CoA because we hooked it up with vitamin A. Alright, we then hooked that 2 carbon to a 4 carbon to make a 6 carbon. Then we took some energy out in the form of bonds, and when we did that, it kicked out those other 2 carbons. Better circle that. Till we get back to a 4 carbon molecule. So now we're going to take a little bit more energy out and convert this 4 carbon back into a usable 4 carbon to pick up some more acetyl-CoA. And that's the Krebs cycle. Should make more sense when we get to the electron transport.